Welcome to this lecture on cell cycle regulation. Imagine your body as a bustling, complex city. For this city to function, millions of cells must grow, divide, and die in perfect harmony. Today, we are going to explore the strict biological traffic control system that makes this possible. We will learn how cells know exactly when to divide and what happens when this delicate system breaks down. By the end of this session, you will understand both the biology and the mathematics behind how your cells protect your genetic blueprint. To understand how our cells divide, let us start with a simple analogy. Imagine driving through a busy city. If all the traffic lights were permanently green, cars would crash, causing absolute chaos. To keep everything safe, the city uses traffic lights that only turn green when it is safe to proceed. The cell cycle works in the exact same way. Before a cell is allowed to divide and create a new cell, it must pass through specific checkpoints. If the cell's DNA is damaged, or if it has not grown enough, the biological traffic light stays red. This stops the division process immediately, ensuring that only healthy, fully prepared cells are allowed to multiply. Now that we understand the need for checkpoints, let us look at the actual path a cell travels. The standard cell cycle consists of 4 main phases. It begins with the G1 phase, which is dedicated to initial growth. Next is the S phase, where the cell synthesizes or copies its entire DNA. After the DNA is copied, the cell enters the G2 phase for secondary growth and final preparations. Finally, the cell reaches the M phase, or mitosis, where the actual division into 2 new cells occurs. The regulatory network we just discussed controls the progression of the cell through each and every one of these distinct phases. So, what exactly acts as the traffic lights between these phases? The regulatory system relies heavily on 2 key types of proteins, cyclins and cyclin-dependent kinases, or CDKs. CDKs are special enzymes that act like an on and off switch for cellular machinery by adding phosphate groups to other proteins. However, CDKs are completely inactive on their own. To function, a CDK must physically bind to a cyclin protein. As the cell moves through different phases, the levels of various cyclins rise and fall. This rising and falling turns specific CDKs on and off, driving the cell forward into the next phase at precisely the right time. This brings us to a critical question. Why does this intricate regulatory system matter so much to our survival? Without cell cycle regulation, cells would divide uncontrollably and endlessly, ignoring the physical boundaries of surrounding tissues. This unchecked, explosive cell division is the exact biological definition of cancer. Furthermore, the regulatory checkpoints ensure that any damaged DNA is repaired before it is copied. If a cell cannot fix its damaged DNA, the regulatory system forces the cell to undergo programmed cell death, known as apoptosis. Therefore, this system is not just about making new cells. It is about maintaining the absolute integrity and safety of your entire genetic blueprint. Before we move on to the mathematics, let us visualize this entire process. On your screen, you can see a circular diagram representing the cell cycle. Notice the 4 distinct phases, G1, S, G2, and M, arranged in a continuous loop. Between these phases, you will see prominent red stop signs. These represent the critical checkpoints we discussed. At each stop sign, you can see that a specific cyclin CDK complex is required to turn the light green. If the complex does not form, or if there is DNA damage, the cycle halts right at that stop sign. This visual perfectly summarizes the strict step-by-step checking process that guards your cellular health. Now that we have a clear visual understanding, let us look at how a cell decides to move to the next phase mathematically. The activation of the cell cycle machinery depends entirely on the concentration of the active cyclin CDK complex. We can model this using a simple chemical kinetics equation. A equals K times C times D. Here, A represents the total enzymatic activity required to pass a checkpoint. The symbol K is the binding rate constant, showing how quickly the proteins bind. The variables C and D represent the cellular concentrations of cyclin and CDK. Because CDK levels remain constant, the activity A is primarily driven by the changing concentration of cyclin. When cyclin increases, activity increases until it hits a critical threshold. Let us see this formula in action with a concrete example. Imagine a cell in the G1 phase that needs an activity level of 500 units to pass the checkpoint. We are given a binding constant of 2 per minute and a constant CDK concentration of 10 units. We need to find the exact concentration of cyclin required. First, we take our activity formula and rearrange it using basic algebra to isolate the cyclin concentration on one side. This gives us cyclin concentration equals A divided by the product of K and D. We substitute our known values, 500 divided by 2 times 10. The denominator becomes 20, and 500 divided by 20 gives us exactly 25. This means the cell must accumulate 25 units of cyclin to flip the biological switch. Now that we know how much cyclin is needed, this leads us to an important question. How long will it take to produce it? We can find this using a simple rate equation. The time, T, equals the required concentration of cyclin, C, divided by the production rate, R. In this formula, T represents the total time required to synthesize the protein. The numerator is the target concentration we just calculated, and the denominator is the steady rate at which the cell manufactures cyclin. Conceptually, this is just like calculating how long it takes to fill a bucket with a steady hose. If the production rate increases, the time required decreases, allowing the cell to pass the checkpoint faster. Following up on our previous calculation, let us find the exact time it takes for our cell to pass the checkpoint. We know from our last step that the cell requires a cyclin concentration of 25 units. We are given that the cell produces cyclin at a steady rate of 5 units per minute, starting from 0. We take our time formula and plug in these values. We divide the total required concentration of 25 by the production rate of 5. This simple division gives us 5. Therefore, it will take exactly 5 minutes for the cell to synthesize enough cyclin to reach the critical threshold. Physically, this demonstrates how cells use the slow, steady buildup of proteins as an internal biological clock to ensure they spend enough time growing. So, we've seen how individual cells time their internal cycles. But how do biologists study this in a laboratory? When we look at a population of cells growing in a dish, they are usually at random stages of their cycle. To understand how much time they spend in the actual division phase, called mitosis, we use a concept called the mitotic index. The mitotic index is simply the fraction of cells in a population that are currently undergoing mitosis. Because the cells are unsynchronized, the percentage of cells we catch in the act of dividing is directly proportional to the percentage of time a single cell spends in that division phase. This is a very common and powerful technique. Let us look at the mathematical definition of this concept. The formula is IM equals NM divided by NT. Here, the variable IM stands for the mitotic index. The numerator, NM, represents the number of cells currently observed to be in the mitosis phase. The denominator, NT, is the total number of cells observed under the microscope. By dividing these two numbers, we get a decimal fraction. This fraction represents the proportion of the entire cell cycle that is dedicated to division. If the number of dividing cells increases, the mitotic index increases, indicating that the mitosis phase takes up a larger portion of the cell's total lifespan. Let us apply this formula to a real laboratory observation. Suppose you are looking through a microscope at a dish of cells. You count a total of 1,000 cells. By looking at their shapes, you identify that 50 of these cells are currently in the process of dividing. We want to find the mitotic index. We take our formula and plug in the observed numbers. We divide the 50 dividing cells by the 1,000 total cells. This gives us a decimal value of 0.05. This means that 5% of the cells are in mitosis at any given moment. Consequently, we can infer that a single cell spends exactly 5% of its total cycle time in the mitotic phase. Now that we have the mitotic index, we can determine the actual real-world time a cell spends dividing. The formula for this is Tm equals Im times T. In this equation, Tm denotes the actual time spent in the mitosis phase. Im is the mitotic index we just calculated, which acts as our percentage. Finally, T represents the total duration of the entire cell cycle. By multiplying the total cycle time by the fraction of time spent in mitosis, we convert our observed ratio into a concrete measurement of time. If the total cell cycle time T is longer, the actual hours spent in mitosis will proportionally increase. Let us finish our laboratory calculation. We found that our cells have a mitotic index of 0.05. We are now given that the total cell cycle time, T, for these specific cells is 20 hours. We want to find exactly how long the mitosis phase lasts for a single cell. We use our duration formula and substitute the known values. We multiply the mitotic index of 0.05 by the total time of 20 hours. 0.05 times 20 equals exactly 1. Therefore, the final answer is 1 hour. This tells us that out of the entire 20-hour cycle, the cell spends only 1 hour actively dividing. The other 19 hours are spent growing and copying DNA, illustrating how brief the actual division phase is. We have covered a lot of ground today, so let us review the most important ideas. First, cell cycle regulation is a vital control system that uses strict checkpoints to dictate exactly when a cell should grow, copy its DNA, and divide, preventing catastrophic errors like cancer. Second, this system is driven by the mathematical accumulation of cyclin CDK complexes, acting as a precise internal clock. Finally, we learned how to use the mitotic index to calculate the exact duration of cell phases from simple laboratory observations. You now have a profound appreciation for the precise, clockwork nature of cellular life. Great job completing this topic.